It's my honor actually to introduce our next speaker, a fellow person from Edinburgh. So it's my honor to introduce actually Robert Illingworth. So Robert have started his academic journey here in Edinburgh. After doing his undergrad in molecular biology, he became a PhD student with Adrian Bert, working on the developing novel biochemical approaches to characterization of CpG island methylations in mammalians. And then after 2 years postdoc with Adrian, he moved to Wendy Bickmore's lab again here in Edinburgh, working on the epigenetics during mammalian embryonic development. And since 2018, I think he has created his own group in Institute of Regenerative Medicine here at the university, where he's a group leader. And we are really honored to have you again as part of Fragile Nucleosome and give you a talk. So let's give a warm welcome to Robert again.
Thank you, Uldus, for that very warm welcome. It's a pleasure to be here, and I'd like to just start by thanking all of the organisers for this opportunity to come and present some of our work. As I said to the organisers when I first came on the call to test out things, that I think they've done a fantastic job of maintaining the momentum of this seminar series post-COVID, and I think it's a great testament to the people who get along, which I'm not patting myself on the back, but I think they've done a very good job. So it's a pleasure to be here presenting our work. I will share my screen. And hopefully you can see my talk. Somebody shout if you can't. My name is Rob Illingworth, as just been introduced, and I established my lab a number of years ago in the Centre for Regenerative Medicine, which you can see in the top of the picture here. And you can tell this picture was taken a while ago because it's sunny. We haven't seen the sunshine since just back into the pandemic. However, I am happy to say, now got an excellent shiny new building to the right of Centre for Regenerative Medicine, as we've now expanded the Institute for Regeneration Repair here in Edinburgh. So it's an exciting place to be. So sales pitch for anybody thinking of moving to Edinburgh or particularly the IRR, please reach out and speak to me. So as Uldus indicated, I have had an interest in chromatin-based modes of gene regulation for the better part of my entire career, and hopefully I can so very little introduction required for this audience, of course, but I have been interested in just about all aspects of this structure. Laser pointer. So I started off my PhD very much interested in DNA methylation CpG islands, we've just been hearing a lot about in Adrian Bird's lab, and then during my postdoctoral career and into starting my own lab, I've had an interest in both the variants of histones that you can incorporate into chromatin, such as H2AZ, the post-translational modifications to histone tails, and for a brief spell a few years ago, also a very real interest in the higher order packaging of chromatin into the nucleus, particularly looking at polycomb-associated structures. And when I set out to start my lab, for a range of reasons, both scientific and personal, I was interested in moving away from looking primarily at the early embryo and looking at the role of chromatin modifications during neurodevelopment. And as I was doing this, I became acutely aware that when you start looking at the human genetics, the underlying causal relationship between the genes giving rise to monogenic neurodevelopmental disorders, there is a substantial enrichment for chromatin modifiers. And here in this slide, all I'm wanting to try and get across is that if you look at, here I've indicated two repressive modifications associated with the polycomb system, two active modifications associated with trithorax, and using as a phenotypic measure of neurodevelopmental disorders of brain growth, arrows pointing down being microcephaly, so a head circumference that is smaller than the average, and macrocephaly with a head size that is larger than the average. What you can see here is that there are extensive abnormalities in terms of brain size in neurodevelopmental disorders associated with both the writers and the erasers of these modifications. And in particular, many of the disorders are associated with microcephaly, so a smaller than average head circumference. And I think what particularly struck me when you're looking at, for example, K4 trimethylation here is that you have microcephaly associated with both mutations in the writers and the eraser of these modifications. The same is true with K36 trimethylation. And for me, this highlights that our understanding at the molecular and gene regulation level does not feed through to a very good understanding of what happens at an organismal level, in the sense that in each of these cases, you affect the deposition or the erasure of these modifications and get grossly the same neuropathophysiciology. So to continue on in the same sort of vein, so this was a huge field, and actually I'm just showing you a snapshot of the history of modifications that are involved in neurodevelopmental disorders. I continued on with a regulatory node that was interested me in the past, and that is the control of H2A lysine 119 ubiquitination and the PRC1 PRW axis of regulation. So by way of background, although I suspect many people in the audience will be very familiar with this, we have E3 ubiquitin ligase complex, PRC1, which can deposit monoubiquitination in this H2A, on the tail of H2A. And the monoubiquitination, for those who are not familiar with ubiquitin, is a 76 amino acid globular protein post-translational modification. And you have the reciprocal activity in the polycomb repressive D-ubiquitinase complex, which can take this very same modification back off chromatin. The two of these complexes work antagonistically with regard to the deposition and removal of this mark. So the work spanning 2 decades now with a lot of people, I've got a list of authors down here, and this is probably 150 of the people I should credit. But what we know is a huge amount about the acrobatics, particularly of this complex, the PRC1 complex. And actually, there are variant forms of PRC1 based on substoichiometric subunits, but you can broadly categorize them into canonical PRC1 and non-canonical PRC1. And this can be functionally distinguished by the fact that PRC1 in the canonical category, through associations with PHC and CBX subunits, are generally involved in chromatin compaction and the interactions of polycomb sites across distal portions of the chromosomes, whereas non-canonical PRC1, through the activating effect of the RYB and YAF2 component, are primarily responsible for the ubiquitination activity. And as I mentioned, the PRW complex, which has got the central catalytic component, BAP1, is responsible for removal of this activity. So what, just to zoom in on this, the reason that other than my background interest in these complexes, was the fact that actually when you look particularly at intellectual disability associated neurodevelopmental disorders with both brain growth and behavioral abnormalities, you find that there are not just one or two, but a whole host of subunits that are part of these two complexes, which are mutated in these disorders. And actually, this list is somewhat out of date now. There's now another example of a neurodevelopmental disorder with a mutation in ring 1B, so the core E3 ubiquitin ligase of PRC1, and there's also another component of the PRW complex. So it seems to be that both disruption of these two complexes are both strongly associated with abnormal neural development. So just to sort of take a slight step sideways at the moment, if we're going to talk about neurodevelopment, I thought I would give us a very quick overview of what I think about when I'm talking about this. So here is a rather complicated schematic of cortical development. I'm talking about cortical development in particular because it is thought that many examples of microcephaly principally come from abnormal growth of the cortex, and also the cortex is largely responsible for the higher functions that we enjoy as humans. And without going into huge amounts of detail, you can grossly break what is a complicated slide here into 3 parts. So we've got developmental time along the bottom. This is mouse embryonic timing. And what you have initially the expansion of neural stem cells in the neural epithelial layer. And these transition to a different type of neural stem cells called radial glial cells. And these, during this neurogenic phase, the neurogenic phase is when you start producing the majority of your cortical neurons. Radial glial cells can do essentially two things. They can either make more radial glial cells, or they can produce the intermediate progenitors and the neurons of the cortical layers, which are starting to build up here, which increases the bulk of the cortex as you go through neurogenesis. And then by about embryonic day 16 to embryonic day 17, you switch from neurogenesis to gliogenesis, where your radial glial cells are repurposed to produce glia rather than neurons. And at this point, you have, if you're a mouse at least, but the same is true, will be on a different time scale for humans, you have the majority of the neurons that you will have for the rest of your life. So obviously, really all I wanted to get across here was this process has to be incredibly tightly regulated to ensure that you have the right amount of stem cells and the right rate and timing of differentiation to produce the correct number of cells to give the brain of the right size, but also the right composition. So experimentally, what has been done, so we know that from a human genetics perspective that polycomb complexes are mutated and give rise to neurodevelopmental disorders. And we know that the neurodevelopmental program is key to ensuring the correct composition and size of the brain. So what has been done experimentally? So in the lab of Haruhiko Kaseki back in 2009, they took ex vivo neural stem cells from mice, and they disrupted either Ring1B, the core component PRC1, or EZH2, the core component PRC2. And what they found was that actually doing this, they extended the neurogenic phase. So the neurogenesis persisted for longer than it did in wild-type cells. In the following year, a similar study, but using an inducible system to disrupt EZH2 in vivo, they actually found, interestingly, the opposing result. They found that actually disruption of PRC2 in this context led to reduction in neurogenesis, as you can see here. And this corresponded to reduced cycling. So the cells, neural stem cells, at this time cycled more slowly. And then several years later, and I think this was in the lab of Lucas Summer, but also in collaboration with Haruhiko Kaseki, when they did an inducible EZH2 knockout in the midbrain using a Wnt1 driver, they found a transition from a midbrain to forebrain-like phenotype. So what I wanted to get across here is what you can see is when you disrupt either PRC1 or PRC2 complexes at different times and in different portions of the brain, that you change the developmental speed and outcomes during this critical window of brain development. So I come back to this slide earlier on. The fact that we see so many subunits of these core components leads us on to the hypothesis, basically, that our lab works from. And that is that essentially the disordered brain development, due to the components of both PRC1 and PRW, operate through a shared mechanism. And then this really boils down to, and this is where my Goldilocks analogy in my title came from, is that you really require the tight control and therefore appropriate levels of H2A lysine 119 ubiquitination to ensure appropriate great brain growth by controlling the balance between neural stem cell proliferation and differentiation. So the lab works on this grossly from two angles. So here are the various members of my current lab, and they split to two subjects. One of those who study the impact of impaired PRC1 function, which are Lucy and Eleftheria at the top, and the other three, who've got Feruze, Charlotte, and Daria, who all look at the contribution of PRW to this axis. But in the interest of time, I'm going to focus on PRC1 for today, and the impact of impaired Ring1B function. So back in 2015, during my postdoctoral work in the lab of Wendy Bickmore, we generated a catalytically hypomorphic form of Ring1B, both in embryonic stem cells and mice. And this operates essentially by disrupting the interface between the E2 protein, which hands the ubiquitin over to its substrate, and the E3, and therefore dramatically impairs the ability of Ring1B to actually add ubiquitin to its substrate. And here you can see the Western blot, you can see the wild type level of ubiquitin. Full Ring1B knockout is undetectable, and what you can see here is the residual amount of ubiquitination in this mutation. And I won't dwell on the fact that also these cells have still got Ring1A, the paralog of Ring1B. What we don't know is how much of this is the activity of Ring1B all the activity of ring 1A in these cells. But what we wanted to do is use this system, as hypomorphic system, we have impaired ubiquitin deposition, to look at the ability of these cells to essentially undergo normal neurodevelopment. So to start with, we wanted to ask a very simple experimental question, which was, can you take these hypomorphic embryonic stem cells, differentiate them into neural progenitor cells, which using a protocol from Austin Smith and Pollard gives you both immature neurons, neural progenitor cells of various types, and neural epithelial cells. So we differentiate our embryonic stem cells in parallel to wild type cells for 14 days, and then we perform RNA sequencing. Actually, here's some examples of what they look like. And I'm not going to dwell on this for too long, but you can see the embryonic stem cells to start with. And what you can see in the wild type, you have these lovely extensive networks of neural projections. You actually also get the same in the I53A, but they're just slightly more dense. Here you can actually see them in the background. But you also have these three-dimensional structures, which I'll come back to in a few slides. So what do the bulk RNA sequencing of these cells look like? So across the top, we have the differentiation dynamics at the level of gene expression in our wild type cells and of our hypomorphs. And what I really just want you to get across here, if you compare day 3, day 7, day 10, day 14 back to the original ES cells, you can see that you get these very dramatic changes in gene expression, both in the wild type, and a very similar extent of changes in gene expression in our I53A hypomorphs. And actually, if you compare these, of course, these could be a completely different set of genes to these, but if you compare these by looking at heat maps, if you take the genes that are upregulated across the differentiation in the wild type, they are grossly the same genes that are upregulated in our mutants. The same is also true for those genes. However, it is clear, and I'm not going to dwell on the genes that I pointed out here, but it is clear that there are some clusters of genes that do not behave as expected. You can say that these grossly make neural progenitor populations, but that there are some gene differences. So what are those? So for this, we now explicitly look to each day of our differentiation to see what are the genes that change their expression. And as you can see, they're not dramatically large numbers of genes, but you can see by day 14, we have about 200 genes that are upregulated and about 150 that are downregulated. And the first rationality, or the first QC step that we wanted to do, was just make sure that these genes are in fact polycomb targets. So for this, we performed Ring1B chip and also RYBP chip. So this is obviously Ring1B is the core component of PRC1, and RYBP is the component, the activating component of non-canonical PRC1 that was largely responsible for ubiquitin deposition. And all I really want to highlight here is that the genes in red that are upregulated in our I53A hypomorphs are preferentially polycomb targets compared to all genes or those that are downregulated. They are also preferentially enriched for RYBP. And if we look, and I won't go into huge amount of detail about this, but if you look, they are also enriched for ubiquitin. So these are the upregulated genes, all genes, downregulated genes, and they have a higher level of H2A ubiquitin at the promoters. So the genes that are upregulated are direct targets in the main of PRC1. So what are they? So when we look at them, these are now specifically the genes that are upregulated in our hypomorphs. You can see that you have a whole host of non-lineage appropriate transcription factors, particularly evident in GATA4 and GATA6, which are important mesendodermal transcription factors. And similarly, if you look at those genes that are disproportionately downregulated in the I53A compared to their wild type counterparts, you see a downregulation of key forebrain and neuronal differentiation markers. If we do functional analysis, this is broadly what we see. So upregulated, we see in particular markers of heart development, but also markers of the mesoderm and endoderm generally. And reciprocal to this, we see a downregulation of forebrain and central nervous system development. So they do make neural stem cells and neural progenitor cells, but they do so less well. So this is the bit that I enjoy the most of the entire talk, because I didn't believe it when I first saw it, but have subsequently been convinced. But as I pointed out in the bright field images I showed you earlier, there were very evident three-dimensional organoid-like structures within our 2D culture, specifically in our I53A hypomorphs. And when I looked down the microscope, as I've now done subsequently a number of times, this is what you see. And you don't really need to be a developmental biologist, because I don't qualify myself as one. but no neural lineage cells should do that. So this is fairly compelling evidence that our gene expression changes that we see in our RNA sequencing data, where we see enrichment of genes involved in both muscle and in particularly cardiac muscle development, are being borne out in our cultures. And actually we see this every time we repeat this experiment with these cells. So what this shows us is that our reduced neural differentiation is counterbalanced by an enrichment of non-lineage appropriate gene expression, particularly of mesoderm and endoderm lineages. So this is all well and good. I'm just checking my time to make sure I'm not, but I'll continue. We're about halfway through. This is all very well and good, but this is a very synthetic in vitro system. So we wanted to move to something that has a bit more in vivo relevance. So for this, we took advantage of one of our next door neighbors, Steve Pollard, who a number of years ago developed a protocol to isolate primary neural stem cells, principally radial glial cells, from the developing forebrain of mice, and it actually works for human fetuses as well, and culture them in EGF and FGF. And what this allows you to do is essentially take neural stem cells that have developed in the embryo until embryonic day 10.5 in this case, and then culture them stably in vitro to then allow you to do molecular dissection on them, which is very challenging to do in vivo. So this is what we did. We took the telencephalons of E10.5 embryos from both our wild type and our I53A mice and profiled the gene expression changes there. So we did this from 2 different litters from 3 different wild type and I53A clones. And what you find is you get a fingerprint associated with our I53A hypomorph, where you have approximately 250 genes which are preferentially upregulated and about 170 genes which are downregulated. And what parallels quite nicely with what we saw in vitro is we see an upregulation of non-lineage appropriate genes, particularly axial development. So that's presumably principally driven by the upregulation of Hox genes. You get upregulation of epithelial cell migration signals, but particularly important, you get upregulation of neural crest signatures. And the counterbalance to this, and very similar to what we see with our in vitro differentiation, is you see downregulation of neural or neuronal function terms, particularly axon guidance and axonogenesis. So this seems to parallel quite well what we saw in our in vitro system. But the thing that strikes me as somewhat strange with this is when you consider that there are in excess of 1,000 genes that are targeted by this system, why do you get a relatively modest change in gene expression? So less than a quarter of the genes that are targeted by this system seem to be affected. So for this, we wanted to actually look to see whether our hypomorph reduces ubiquitination globally across the genome everywhere, or if some genes preferentially lose more than others. So for this, we performed calibrated chip sequencing for H2A K119 ubiquitination. And this is just an example, a zoomed out example of chromosome 1. And what you can see in the back here is the wild type levels across the chromosome of ubiquitin. And here in our hypomorph, you can see the levels are reduced. And this is just a quantification of that. And we can see that in neural stem cells, in these cultured neural stem cells, we're getting a reduction of about 30% of wild type levels. This is obviously very zoomed out. We're looking at essentially 200 megabases of DNA. So if we zoom in, what do we see? So here we have the GATA4 locus, the gene I mentioned earlier on, and engrailed on the right-hand side. And what you can see in our I53As, we consistently have a lower, quantitatively lower ubiquitin signal, and engrailed it's a little bit more pronounced. But is this reduction true of all genes? So this is actually just a meta-analysis of all genes. These are not enriched for polyclonal genes. All genes, you've got wild type here and our hypomorph here. And what you can see is there is a reduction. This is represented here in this profile, a reduction globally across all genes. But is there a bigger effect, or is there more signal if we look specifically at those genes that are upregulated? The simple answer to this question is yes. So what the genes, not surprisingly, that are upregulated have a higher level of H2A lysine 119 ubiquitination. But what is clear from this is, whilst these ones have more ubiquitin, the magnitude of loss, the actual ratio of loss, is not that much different to all other genes, irrespective of whether they're upregulated or not. So what you can see here is just a quantification of this. This is comparing wild type to wild type here, hypomorph to wild type here, and a different hypomorph to a wild type here. And this is the ubiquitin signal of gene promoters. In red are those genes which are upregulated, and in grey are all of the other ones. And really all I want to get across from this is that actually the vast majority of genes who lose their ubiquitin levels in these clones do not upregulate their genes. Now, why this is, I have not got a definitive answer for you today. This is something we're currently working on, but the preliminary data that we've generated gives quite a simple explanation. And that is essentially, you remove a global repressor like this in a given context, in this case in neural stem cells, and you only activate those genes that you can activate, i.e. the ones that have got transcription factors that bind to those promoters in the media conditions and in those cells at that time. So the suggestion here is that essentially the genes in red are those genes that can be turned on because the transcriptional machinery is present. We're doing some final experiments to test that, but that is the likely, I think, conclusion to this experiment. So this is very good. So what does this mean for the developing mouse brain? So obviously, because we have the mouse line, we can actually look at what the developing cortex of these mice look like. So on the left here, we have 2 wild-type E12.5 mice, and on the right, we have 2 I53As. And you can see that grossly developmentally, they're normal, particularly when you compare these to ring-1B knockouts, which don't make it past gastrulation. They're slightly developmentally delayed, but in many instances, what you can see is the cortex is smaller. In extreme cases, we've seen examples where the cortex is actually slightly concave. So what do these look like if you look at them down the microscope? So here we have taken a cross-section across the cortical to ventricle layers. So say this is very early on as you just start to move into neurogenesis. And what we can see is if you orientate yourself, this is the cortical surface, slightly counterintuitively the outside of the brain. This is the ventricle, so the inner side. And what you can see, stained in SOX2, are in wild type, you've got this population of neural stem cells. Here on the other face, as you go to the cortical surface, you can see your tubulin positive neural projections. And you can see you get this lovely pattern. So you go from the stem cell population to intermediate progenitors and immature neurons. What do the I53A look like? Well, what you can see, hopefully from this, although this is not the best section, you can see that the SOX2 positive stem cell population is diminished and the TUJ1 positive population has been expanded. And we see this consistently across our embryos, and this rather suggests that we are losing the key population of cells, or at least reducing the capacity to maintain this population of stem cells, which is then critical to build the later cortical layers. So we are currently doing more in vivo cell biology work on this to try and get a better idea of what is actually happening. Are we getting reduced cycling dynamics? Are we getting enhanced differentiation? Or as our ex vivo analysis suggests, are we actually also at the same time producing ectopic cell types, possibly of the neural crest, that we should not be seeing at this point? So that's work ongoing. So I will just run through the summaries for this part of the talk. There's only 2 more slides to go, don't worry. So what we can see is when we take our hypomorphic ring1B cells and try and differentiate them into neuroprogenitor cells, we can make them into neural progenitor cells. However, they do so with lower efficiency and also with skewed developmental trajectory, with the appearance of ectopic expression of non-lineage appropriate PRC1 target genes, particularly those of the mesodermal and endodermal lineages. When we look at our ex vivo neural stem cell culture, we can culture out these cells in EGF and FGF. So these are conditions which maintain radioglial cell populations. They're stable long term, but they show a generalized upregulation parallel to what we see in vitro of PRC1 targets. And similar to what we see in vitro, we see a reciprocal downregulation of neuronal gene markers and upregulation of non-lineage appropriate genes. This corresponds to the fact we see the global reduction in H2A lysine 119 ubiquitination, but that interestingly, the genes that are upregulated do not show a substantially higher level of reduction of this modification, suggesting that actually we're only upregulating a subset of genes that can be upregulated. And this ultimately in vivo is borne out by the fact that you seem to have a reduced population of neural stem cells in the E12.5 developing cortex. So actually, in the interest of time, because I realize I'm out of it, I will largely skip past this. But as I indicated at the beginning, we also have an interest in the counteracting activities, the PR-dub complex. And this is really due to the fact that there are very, very strong evidence now that the various components of this complex, and here I focus on the ASXLs, that when you mutate them, you get a range of profound monogenic syndromes that affect or give you systemic wide phenotypes, but they give you very clear brain growth abnormalities. We're actually trying to understand this experimentally, as this is part of collaboration with Frida Miller and David Kaplan in UBC to do spatial transcriptomics to start profiling the expression of these components. But I will not dwell on that anymore, and I will cut to the end. And thank you, first and foremost, for your attention, for that whistle-stop tour through this unpublished work. But also thank my lab. I'm very lucky to have this great junior lab that are supporting our research. And in particular, the work that I presented today was by Lucy Doyle, Dipta Sengupta, who now works here at the CRM, collaboration with Ian Adams and a former research associate, Alexandra Darian, as well. I will stop there, and if there's anybody got any energy left for questions, I would be delighted to answer them.
Actually, we have many questions, so like 3 in the Q&A and 3 hands raised. So I will start with Q&A. So Jin Young Park asked if ring 1b mutants make the cell to differentiate into mesoderm or endoderm. Generated cells are normal, or do the cells have mixed cell identity of mesodermal cells and MPC?
So it's a fantastic question, and one I wish I could give you a definitive answer to. So actually, we have done very similar experiments to these as part of collaboration with Josh Brickman, where we look at definitive endoderm differentiation. We see exactly the same phenomenon. We get dysregulation of the developmental trajectory and ectopic expression of other cells. And in both instances, it looks like we're getting production of cells which look like they're biological in vivo counterparts that aren't chimeric in terms of being endodermal and mesodermal, or in that case, mesodermal and ectodermal. But we have yet to do the killer experiment, which is either IF or FACS or a single cell. But these are experiments we're going to do, so that hopefully next time I can answer your question. Okay,
so James Gunn asked, what effect does I53A have on K27 prime methylation levels in this context?
It's a great question. So in embryonic stem cells, where you have a much more pronounced loss of ubiquitination globally, you get, and that was the 2015 paper, you get a parallel reduction in K27 trimethylation levels, and it scales with the reduction in ubiquitination. Here, where the ubiquitination level, the reduction, sorry, in mutation is rather more modest, you see a reduction in K27 trimethylation, but only at those sites where you have a large amount to start with and a reduction of about 50%. Any less of a reduction than that, you do not see a reduction in K27 trimethylation. The confounding factor, if I may add one other note to that, is the fact that generally where these biggest changes happen are more often than not where you get gene expression. So we can't unpick the change in K27 levels from the inability to place PRC2, from it being driven off chromatin due to activation of gene expression. But there are a subset where you see a large difference in ubiquitination and they lose K27 and they aren't activated. So I think the mechanisms of PRC2 recruitment probably are similar to what they are in ES cells.
So I actually, Pradeep has a question and I added him so he can ask. Pradeep, you can unmute yourself. Yeah, you have unmuted.
Hi, can you hear me?
Go, Pradeep.
Yes, Pradeep. Nice to hear you.
Yes, a fantastic talk, Rob. I had slightly confused between in vivo and your in vitro data. That question I'm going to ask you later because it may need more time to answer. One question I'm just trying to pick your brain about is how common it is when you perturb a epigenetic regulator in a in vitro differentiation system, or even the mouse model, or in the organoid models, to see a kind of a microcephalic kind of phenotype where you see increased 2G1 positive cells and reduced NPCs, and then you kind of start seeing a smaller brain size. Because like kind of seeing that anything you perturb, you start seeing that I see your patient data, we see a microcephaly is quite predominant in many patients with mutations in neurodevelopmental genes implicated in NGDs. I'm trying to understand, what makes this a very kind of a common phenotype.
I don't have a very good answer for you. I would say I agree. It seems to be that if you mess, or at least in for the regulators that I look at, so mainly chromatin regulators, it seems to be when you disrupt them that you get, grossly speaking, a poorer ability to maintain your stem cells, or the stem cell cycle slower, and presumably as a result of that, you get disproportionately a higher proportion of immature or intermediate progenitors. In terms of whether you can get a measurable microcephaly phenotype, I have only ever heard one person talk about that, and it wasn't a chromatin regulator, but actually John Mason, who I think is actually on the call, and David Price have looked at this in human organoids, and actually got a beautiful measurable reduction in organoid size in a model that gives you microcephaly in humans. But as for what you see when you're looking in vitro systems, it does seem to be that this is the common output. I would argue that that does give you some indication of the mechanism that you get the disorders in humans, but actually that's possibly because that's what I want to say.
Yeah, good answer. I was wondering, is there anything to do with which we don't really explore, like a cell cycle or DNA damage response, those kind of pathways, because we all look into gene expression, chromatin, when there's these mutations.
So I had a slide in this talk until about an hour before we presented it that showed the I53A mutation, hypomorphs in the neural stem cells have a beautiful reduction in S phase with a compensatory increase in G1. And I removed it because, having literally got some new data in this week, there is one I53A clone that doesn't do the same thing. So in all the cases where we see a reduction in S phase, it seems to be we get upregulation of the INK4R flocus in our hypomorphs. But in the one that doesn't, it doesn't seem to be the case. So it is not a fully penetrant phenotype, but it is a polychrome target, and that is to do with cell cycle regulation. So I think that is a common phenotype, but we haven't done enough clones to nail that down, and I've removed it from the talk because I can't say anything more robust than that.
Thank you.
So yeah, thank you, Pradeep. So Carmela, I will allow you to talk, but if it's a short question, because there's also a Nirban, and I want to let him talk too.
Hi, Rob. Very nice talk.
Hi. Thank you.
So I have a question still on the same line of the K27 trimethylation. So do you see that the genes that are basically more upregulated or that respond more efficiently, do they have any kind of special kind of distribution? I mean, what we have seen is that normally these very important genes that actually, by the way, we found that there were genes that were poised also in terminally differentiated neurons. And one of the striking points, it was that there were master genes for cardiac differentiation. So do you know if these genes are particularly bivalent, for example, and if they have a broad distribution of the signal all around the regulatory sequence? Because these genes, they were having many enhancers close by. So it could be that they are particularly susceptible to have a very strong burst of response.
I haven't done this. So the genes that are upregulated, as I indicated before, tend to have a quantitative, an obvious reduction in K27 trimethylation. I haven't done a spatial analysis to see whether they look different in terms of the domain size compared to genes that are not. Actually, it's part of, so all the data is processed to do exactly that type of analysis in terms of also motif enrichment analysis in the vicinity of the promoters and so on, to try and rule in or out what might be leading to the upregulation of this subset of genes versus the others. So the simple answer is I can't tell you, and I don't think I've looked enough at the browser tracks to even give you an indication. But it would certainly, the master regulators that are coming up are obviously very tightly developmentally regulated. Therefore, they will likely have complicated cis-regulatory elements within the vicinity of the promoters. Therefore, I could very much believe that that would be the case. But I can't answer your question definitively.
Yeah. Thank you, Carmela. Sorry, I need to make other people also ask questions. So Julian, you have a question, if you can go fast.
Yeah, great talk. I was wondering whether the patients with this hypomorph mutation have like cardiac cells where they're not supposed to. Are you able to look at that?
So there's two things. This is not a disease-causing mutation, so we're using this very much as a model to look at perturbed ubiquitination rather than modeling a disease-causing mutation itself or syndrome. It's also, I should probably say, it's probably more of a dramatic mutation than most of the other ones that you'll look at. So there's only, to my knowledge, one Ring1B mutation that's associated with neurodevelopmental disorder, and it has not been characterized at the molecular level, but I would suspect it does not lead to such a pronounced effect. The second part of that is I would doubt it. So from this data, and actually a lot of data that I haven't presented today, I think generally speaking, what you see is you make lineages that are completely inappropriate if you start disrupting your epigenetic system much earlier. I think the more constrained the environment in which you're differentiating, the more restricted the ectopic outcomes can be. So for example, in our neural stem cells, you get a less broad range of abnormal lineages, I think, because they are actually developed in the confines of the actual developing brain, which means there will be signaling and physical constraints to what those cells can become, and therefore they don't become heart in that context. So I think it's probably to do with constraint, whereas if you start with embryonic stem cells, they can obviously sample essentially all the developmental lineages that sit in front of them, and therefore you can get heart in that context. That being said, we will have a look for some cardiac markers just in case I'm wrong.
Okay, thanks, Julian. Now, Anirban, I have unmuted you if you can ask your question.
Yeah, can you hear me?
Yes.
Okay, great presentation, Robert. I think somebody already touched base with a similar question as mine. So pardon me if I'm being repetitive. I was wondering if the cortical development impairment that you see in the catalytic deficient Ring1B can be mimicked by messing around with other subunits of PRC1 complex, or maybe some of the enzymes that mediate H3K27 trimethylation. So how specific do you think the contribution of this Ring1B towards this drastic in vivo effect?
I don't think it's specific. I would say is a simple answer. So we are doing this exact sort of series of experiments. So if we go back to my hypothesis is that I believe that probably if you disrupt this axis by any means, that you will get a core set of genes that will change in a similar manner, even if you disrupt PRDUB or PRC1, would be my hypothesis. But certainly, I think if you focus in on PRC1, I think probably anything that you do to perturb certainly non-canonical PRC1 function, I think you will get grossly a similar phenotype. We're testing this in the lab now.
Thank you.
Thank you.
Thank you, Anirban. Thank you for this great discussion. We are slightly over time, but it's okay. It was really nice discussion and really good questions. And thank you, Rob and Julian, for 2 amazing talks today. And hope to see you guys in 2 weeks. Yeah.